Hello, everyone, and thank you for joining us today for another ACE Spotlight Series presentation. My name is Rouzbeh Afrasiabi, and I will be your host. Today, we have Wengong Jin from MIT, who will present his work titled Hierarchical Generation of Molecular Graphs Using Structural Motifs. Wengong is a PhD candidate at MIT, advised by Professor Regina Barzilai and Tommy Yakola. His research interests lie in deep generative models for graphs and their application in chemistry and biology. He's an NVIDIA Fellowship finalist and a recipient of William A. Martin Award for Best Master Thesis at MIT. With that brief introduction, and without further ado, I pass the mic to Wengong to talk about his work.
Thank you. Thanks for the warm introduction. So I'm Wengong, and today I will talk about some of my recent work on the generative models for molecular graphs. So molecules are richly annotated graphs. Here's an example of a antibiotic compound. As you can see, it is a graph where each node is an atom labeled with its atom type. Each edge is bound, labeled with its bound type. Molecules are also composed of substructures or motifs. Certain groups of atoms are called motifs, which have a specific chemical meaning. Together, atoms are called motifs, which give rise to a variety of chemical properties such as solubility and toxicity. There are two important tasks associated with molecules. First is property prediction. Given the input molecular graph, we want to predict what is its chemical property, such as solubility. This task has been extensively explored recently with the rise of graph convolutional networks, or MPNs, and it is relatively an easy task. Another task is generation of molecular graphs, which is the inverse problem of property prediction task. The goal is to generate molecules that satisfies specific chemical properties. For example, here we want to generate an antibiotic compound that kills certain bacteria. And we want, our specification is the molecule to be soluble and also potent against the bacteria. So generative models are very useful for drug discovery, but there are a lot of challenges that remains, even though there has been a lot of work that trying to solve this task. The main challenge of molecular graph generation is that molecules can sometimes have complicated structures, and the properties may depend on intricate features. For example, here you have an antibiotic with a complicated ring system. You may also have a polymer-like molecules where it has a lot of atoms, and it also has complex structures. So previous methods for molecule generation can be roughly divided into two categories. For example, we have atom-based methods like hgraph2graph or graph RNN and more. These methods generate molecules atom by atom. Each step, the model will expand the molecule by one atom. We also have substructure-based methods like my previous work on junction tree VAE. It will, in each step, the model will expand the molecule by one substructure. However, back then we only considered small rigid substructures like small cycles or single edges. So while these methods have demonstrated some level of success in small molecule generation tasks, however, when we apply these methods to generating larger molecules, such as polymers, we found that all methods had really low reconstruction accuracy for larger molecules. So here we tested my junction tree VAE and also a hgraph2graph, which are considered as current, the state-of-the-art models. And as you can see from this curve, these methods typically fail for reconstructing larger molecules that is over 100 atoms. This is quite frustrating because most of the polymer structures are kind of large, and reconstruction accuracy is really important is an important measure for the VAE model performance. And I will show in the later slides that reconstruction accuracy is not the only issue. These two models also have poor sample quality. So let's see what is the reason behind these failures. Let's look at this molecule for a second. This is taken from a polymer dataset from some chemistry journals. So to generate this compound for CGVAE based on atoms, it will take 70 atom predictions plus more than around 150 bound predictions. Since CGVAE is an autoregressive model, during training, you will need to propagate the gradient through this long sequence of generation steps. This imposes a lot of challenges, such as vanishing gradients and learning stability. Same thing also applies to junction tree VAE. Even though because it utilizes only these small substructures like 5 or 6 member rings, it will also take a lot of steps to generate this compound. And that's where the errors get accumulated, and that's why these methods tend to fail to reconstruct large molecules. Our solution is to utilize these larger, more flexible building blocks, which we call motifs. Back then, junction tree VAE only considered single rings and bounds as building blocks. But larger molecules, such as polymers, exhibit clear hierarchical structure being built from repeated structural motifs. For example, you can see there are 2 structure motifs that get repeated in this polymer-like structure. So when generating this molecule motif by motif, you will see by using these larger building blocks, it will only take 11 steps to generate this polymer structure. And the model becomes much easier to learn, and also it is faster. Just to illustrate, since there are a lot of concepts here, just to illustrate what is the distinction between substructures that are introduced in junction tree VAE and motifs in our recent work. So in junction tree VAE, a substructure has to be a ring or a bound or a single atom. But in our work, we consider motifs to be much larger and also to be very flexible. It can be a fusion of many rings or a combination of bounds and rings. So to generate molecules based on these large motifs, we will need a new generative architecture. Unfortunately, previous junction tree VAE cannot be directly extended to use motifs because it requires a combinatorial enumeration to assemble substructures in each neighborhood. And the time complexity is exponential to the motif size. For example, here, this is just one step of the junction tree VAE decoding. It will, after you have predicted all the motifs or substructures, you will need to assemble all these things together, and it will invoke a combinatorial enumeration. And it's easy to have more than 100 different ways to assemble all these motifs together. So with larger motifs, the number of these candidate assemblies will definitely get explode. So this leads to our new model architecture called Hierarchical Graph VAE. It has 3 components. We will first extract motifs from the training data. So here, I will start with a simple motif extraction based on some simple heuristics. And in the later slides, I will discuss how the motifs can be learned based on chemical properties. The second component of our model is this hierarchical graph encoder, which represents molecules at both motif and atom level. It is designed to match the decoding process. The hierarchical graph decoder will assemble motifs one at a time, and it is scalable to larger motifs. So let me go through these, elaborate each of the components in more detail. So first, let me briefly talk about this motif extraction. What we did is quite simple. So here, given a molecule like this, I will first find all the bridge bounds like this, such that either one of the atom is part of a ring. I will detach these bridge bonds from its neighbors, so the molecule will be chopped into a set of disconnected components. I will select all the components as motifs if it occurs very frequently in the training set. Here we set the frequency lower bound to be 100. So if a component does not appear more than 100 times in the training set, we will further decompose it into basic rings or bounds, just as the junction tree VAE. Just to note as a side remark, this is a simple heuristic-based motif extraction algorithm, and it is up to the users to design what are the motifs that they care about. And this motif extraction algorithm can be adapted to different data sets based on the prior knowledge. And in the later slide, I will discuss how these motifs can be learned through some guided by some property criteria. But let's just go with this motif extraction paradigm. So as an additional piece, as you can see, the motif decomposition tend to lose a lot of atom level connectivity information. So notice that when you decompose these motifs, you can actually remember or label these attaching points in the motifs so that you don't lose too much atom level connectivity information. And this will allow us to easier to reconstruct a molecule, and this additional information will be very useful for generation. So by saying marking attaching points, I will, if you can see here, these gray boxes, these will be part of the label in this motif. So just to be more clear, after the motif extraction step, we will end up constructing a motif vocabulary from that's constructed from the training set. So the vocabulary size is usually small. Of course, it depends on the data set. But in our case, we found this to be usually lower than 500. So this is an illustration of the vocabulary of motifs. Each motif also have a vocabulary of potential attaching points. So for example, given this motif, you will see there are 3 different attaching points that occurred in the training set. We will gather all these possible attaching points as the vocabulary for each motif. And we found this is also typically small, usually less than 10 different attaching point configurations.
Wengong,
now,
if I can stop you for a second, we have a question online.
Okay.
They're asking, the motif extraction seems like subgraph matching when checking frequency. Is that not a large computational problem, or is there a shortcut? I'm not so. Oh, I see. No, there. So it actually does not. How can I say? Oh, I see. Yes. So it does involve substructure matching, but we found this to be very fast actually, because we can just convert all these. So let's say I have a molecule, I can chop these into motifs, and I can just get the canonical smile strings for each of these motifs, and I can just do a quick lookup in the vocabulary by just a string matching. This is a pretty fast process. I hope this answers the question. Yeah,
is that okay?
Yeah. Thank you.
All right, thanks for the question. So so now let me talk more on our encoder decoder architecture. This is more, this is the majority of where the deep learning stuff comes from. So just to help you understand our architecture design, let me briefly go through our generation process. So during generation, we will, so our model is autoregressive. In each step, it will try to expand the molecule by one motif. And during generation, we will maintain all possible positions to which new motifs will be attached. As you can see, these are highlighted by these gray boxes in the figure. So each generation step contains three predictions. One is, the first step is motif prediction. So it will predict what is the next motif that needs to be added to the molecule. After we have this motif, we will need to know how to attach this new motif to the current graph. To do this, this brings us to the second prediction, which I call attachment prediction. I will try to predict what are the attaching points in this new motif. And then as a final step, I will predict how these new atoms, or how these attaching points should be connected to the attaching points in the current graph. This is also a classification task. And once you predict the right matching attachment configuration, you can merge these new motif with the current graph, and this will transform the current state into this new state. This formula kind of at the high level describes our autoregressive process. Here, Z is a latent variable in our variational autoencoder. SK, it stands for the case motif that has been generated, and AK stands for this how it should be attached to the current graph. And as you can see, the prediction of the current of the new motif and its attachment is depend on the previously generated motifs and its attachments. So just as a side remark, I want to briefly mention the key distinction between our model and the junction tree VAE. The junction tree VAE will first predict all the motifs using the tree decoding process, and then figure out how to assemble all the predicted motifs. And the decoder will try to assemble each motif with all the neighbors, with all its neighbors in one go. So the combinatorial enumeration is complex and slow, and it's hard to scale to larger motifs. In contrast, our hierarchical VAE decomposes this assembly process into multiple baby steps. We first predict the attaching points, and then learn how to match or attach these attaching points to the current graph. And we also only assemble one motif at a time, not the entire neighborhood. And this we found to make our model much more scalable and faster to handle larger molecules. So now let's move on to the architecture details of the graph encoder and decoder. So to support this generation process, we need to design a suitable encoder that captures all the information needed for each prediction step. In particular, our graph encoder will learn a representation, will learn to represent a molecule by a hierarchical graph that has 3 different layers. Each layer corresponds to each of the 3 prediction steps in the decoding process. So here, the first layer is this atom layer. It's basically the same as the molecular graph of the molecule. It is designed for the graph prediction step because it involves how to merge different graphs together, and it will definitely need this fine-grained atom-level connectivity. The second layer is this attachment layer. It is designed for the attachment prediction step that I just mentioned. So here, each, you can think of it as a graph pulling on top of this molecular graph. Each node here is a motif along with its attaching points. Two motifs are connected by an edge if they intersect. And this layer is crucial for our decoding process. We also have on the third layer, that is motif layer. Here, I remove all the attaching point information. Each node is just a motif, and two motifs are connected together if they have intersecting atoms. This layer is designed for the motif prediction step that I mentioned earlier. As you can see, this is basically the inverse of our decoding process. To encode this hierarchical graph, we run message passing networks on each of the layers. For example, I will run the message passing network on the atom layer, a message passing network on the attachment layer, and a motif layer. So this, in addition, while I'm running message passing on the attachment layer, I will also propagate the corresponding node vectors from the atom layer to this layer. So for example, let's consider this motif here, this blue box. It will not only receive the messages from the neighbors in this attachment layer, but also the messages from the atoms that belongs to this motif. So this will be all the atoms in this gray box. So we call this hierarchical message passing because because the messages are passed along in a hierarchical manner. So our hierarchical graph decoder also utilizes the same hierarchical representation. The generation process can be regarded as incrementally constructing the hierarchical graph. For illustration, let me repeat this one-step generation process that I described earlier. So the first step is motif prediction. It is a classification task to predict the right motif from the vocabulary. After this, I can predict what is the right attachment from the motif from the attachment vocabulary of this motif. This is also a classification task. And then I can predict how this new motif should be attached to the current graph. This is also a classification task because we already have all the attaching points highlighted in the new motif and the current graph. So this is our kind of a basic overview of our generative architecture. So we apply this model to our experiment on polymer generation. Here we took a polymer dataset from this paper, which contained 86K polymers. We divided them into 76K for training and 5,000 for validation and testing. We adopted a variety of evaluation metrics. We first considered the reconstruction accuracy. Basically, we encode a molecule into its latent representation and then learn to decode it back to the molecule. We also measure the validity of the sampled outputs. So basically, we count how many of the 5,000 molecules sampled from the model are valid. We also count the uniqueness, basically saying among these 5,000 molecules, how many of them are unique. We also compute the diversity of the sampled molecules by computing the tanimodal distances between different molecules. In addition, we also try to measure whether the generated molecules have similar property distribution with the two molecules in the test set. So here we computed free shed distance between the property distribution of the molecules generated and also the molecules in the test set. In particular, we compute the log P, QED, synthetic accessibility, and the molecular weight of the generated set and test set. In addition, we also wanted to measure whether on the structural level, the generated molecules are similar to the two molecules in the test set. Here we can measure the nearest neighbor similarity, the fragment similarity, and the scaffold similarity between generated set and test set. More details are mentioned in the paper. So here, as you can see from this table, our model on the bottom, hierarchical VAE, has a much higher reconstruction accuracy, and also it has better lower property distribution from between the generated set and the true molecules observed in the test set. And these are some sample molecules that I generated that I sampled from my model. We also, yes.
Sorry, if we can go back to the previous slide, just for better understanding of the audience, you're comparing SMILES and graph methods. Could you explain to the audience why SMILES methods have usually lower results compared to graph methods?
Right. So SMILES-based methods, how can I say? So first of all, the semantics in a SMILES string is usually not clear, or at least kind of blended because of the additional grammar that utilized by the SMILES string construction. So for example, maybe I have pointed this in my junction tree paper. Two molecules may be similar in terms of its graph connectivity, but it may result in very different SMILES strings. So that's why it is hard for molecules, hard for the SMILES string-based method to learn a good latent space representation. And also, SMILES strings it is order dependent, while the graph, each molecule can result in many different SMILES strings. Even though here in this experiment, we only use the canonical string representation for each molecule. But I suspect this also brings additional challenge for learning these SMILES-based methods. But as you can see from this table, even though it has a low reconstruction accuracy, the property distribution, the property statistics and the structural statistics is not too bad compared to the graph-based methods. So I don't want to bring down the SMILES string methods too much.
Okay, thank you. Another question in regards to processing power required for running the two different models. Can you make a comparison?
You mean between SMILES and graph-based methods?
Yeah.
So yes, indeed, SMILES-based methods tend to be much faster than graph-based methods because graph-based generation models need to each hidden state of the generation process is a graph, while for SMILES string it's basically a prefix of the string. And in that regard, yes, SMILES graph-based method is typically slower. For my model, it is about 10 times slower than the SMILES-based methods on these large polymer datasets. For smaller molecules, this will be smaller, I guess, but even for hierarchical VAE for our model, we can train over 1 million compound in just 3 or 4 hours. So I wouldn't say this is too bad compared to SMILES strings.
Okay, thank you so much.
Okay, so yes, I indeed bring up this speed issue. As you can see here on the far right, this motif-based method does actually train a lot faster than atom-based or these structure-based methods because we generate molecules based on motifs, and we require much less steps to reconstruct a molecule. And this is a great advantage because typically graph-based methods tend to be slower. As you can see, for larger molecules, our model actually still maintains a high reconstruction accuracy, even for molecules with more than 100 atoms, while for other methods, it tends to decrease a lot. In the middle, I have shown this figure. It's basically showing what are the motif size in this data set. We can see there are actually many motifs with around like 20 atoms. So this really means that it's important to incorporate these large motifs in our models. We also have de novo molecule optimization task. Here we aim to transform given molecules into molecules that satisfies given design specifications. This is this is related to lead optimization in drug discovery. In many real-world drug discovery tasks, we are often faced with molecules that possess some good properties, but not perfect. It lacks some other properties. So, for example, we may want to modify this compound to be more drug-like while remaining its potency, or while maintaining its potency. We may also want to modify this compound to be more soluble, or to have a higher log-P while remaining potent. So the task of de novo molecule optimization seeks to learn a molecule-to-molecule mapping, or in other words, graph-to-graph mapping. The output compound is constrained to be structurally similar to the starting point. So this motivates us to formulate, this optimization task as a graph-to-graph translation, which I have mentioned this in my previous work on iClear. So given a source molecule X, I will use an encoder decoder to model to map X into a target molecule Y. The translation model is trained on a set of source target pairs collected from the data set. So the data set here comes from my previous paper. So here we wanted to test whether our model can excel in this graph translation task or not. And it is easy to modify this hierarchical VAE into a translation model by just adding attention layers between the encoder and the decoder. We tested on two property optimization tasks. One is to optimize the drug likeness measured by the QED. Another is to optimize the output molecule's activity against dopamine type 2 receptor. The evaluation metric is success rate. Basically, how often can we succeed in translating input molecule X into an output Y such that they are similar enough, and also Y is drug-like or DRD2 active. So here are some of the results. As you can see on the slide, on this DRD2 optimization task, our model tend to have a much higher success rate compared to the previous methods based on SMILES, junction tree, and also these atom-by-atom based methods. We also observe similar improvements on this drug-likeness optimization. Our model on the far right does perform the best among all these baselines. Okay, just as a sorry for going a little bit over time, but I just want to mention this motif extraction. Back then, I just want to briefly address the problem of motif extraction. Back then, we used some simple heuristics to extract motifs, but this is not really satisfactory. A question that I asked is, can we learn to extract motifs guided by some criteria? For example, I can perhaps extract motifs that are likely to be responsible for certain properties. For example, this motif here contributes most to the inhibition against a biological target JNK3, or I can try to find motifs that inhibits another target GSK3 beta. I just want to say using these motifs that are highly active or highly inhibitive to some of the proteins, using these motifs, we can easily generate these JNK3 or GSK3 inhibitors. For example, to generate a DRD2 active compound, I can start with a motif here that is predicted to be highly active to DRD2, and I can expand it to a full molecule by filling in the missing parts using graph generation. Motifs should have high predicted activities against DRD2. So here we will use a property predictor that will be trained on the dataset with active and inactive compounds. So this sort of brings us opportunities to utilize these motifs for multi-property optimization. You know, drug discovery always involves multiple properties. Therefore, the previous experiments on de novo molecule optimization are only simplified settings for drug design. And multi-property optimization is really challenging. Many examples of molecules, in a typical scenario, we will find that we have a data set with many examples of molecules with just having a particular property just only satisfies one of the single constraints. But we have 0 or very few instances of molecules that satisfies multiple property combinations. As you can see here, in my data set, what we found is we have many JNK3 inhibitors, GNK3 inhibitors, but we don't have these dual inhibitors that inhibits both proteins. That is actually very critical for treating Alzheimer's diseases. So the challenge here is, how do we realize a diverse distribution of molecules that satisfies all the criteria without examples of such molecules? So we can actually use these extracted motifs to generate molecules. For example, to generate a dual inhibitor towards these two proteins, I can extract motifs for JNK3 and GSK3, and I can combine these different motifs and learn to connect them into a full molecule by a graph generated model. With this approach, it is more likely to generate molecules satisfying multiple properties. This is analogous to importance sampling that we typically use for sampling rare events. So now the question is, how do we find these property-specific motifs? So our goal is to, given a molecule G that is active or inhibitive to some biological targets, we want to find a minimal subgraph S such that S has desired inhibition or activity. For example, given a molecule here, we want to find that this minimum core structure that contributes most to the property. So this is combinatorial optimization task, and we propose to solve this using a Monte Carlo tree search algorithm. So here, if you can see from the right, at a high level, we will iteratively remove these peripheral bounds or rings that if the remaining subgraph still have a high predicted property score. And I can repeat this process until that I reach some cores that I cannot break them further. So I will use a property predictor to assess all these subgraphs, and I will select these minimal subgraphs that has high predicted properties as my motifs. So now I can construct the vocabulary of motifs using this Monte Carlo tree search process, and I will use this motif vocabulary to generate molecules. To be more specific, the model will be trained with reinforcement learning. In order to generate molecules that is inhibitive to both targets, I will sample motifs from the motif vocabulary of JNK3 and the motif vocabulary of GSK3, and I will sample one motif from each, combine them, and then complete them into a full molecule. The reward is basically whether this full molecule inhibits both JNK3 and GSK3, and I can train the whole model using reinforcement learning. So compared to previous methods, this model is advantageous because it alleviates this reward sparsity. We found that previous methods using reinforcement learning, it is typically hard to get positive rewards given these multiple constraints. And here the motifs are analogous to options in the reinforcement learning literature that is typically used to address this reward sparsity issue. Just to briefly go through some of the experiments in this paper, in the task of generating dual inhibitors against GSK3 and JNK3, we found that we tested our model and compared with previous state-of-the-art reinforcement learning methods, such as GCPN from Stanford and also reinvent from AstraZeneca. And we found that our model outperforms them with a significant margin. In particular, the output molecules from our model is much more novel than the previous methods, and also it is more diverse compared to the previous models. If we move on to 3 or more constraints, we found that many of the reinforcement learning methods that typically fails, and our model still remains high success rate and novelty and diversity because we're utilizing motifs. And we found that, as you can see from the chart here, our model can still remain a high success rate when you incorporate more and more constraints. So just as a summary, we in this talk, I described how to generate molecules using structure motifs for various domains. We can extract motifs based on simple structural heuristics, and also extract motifs based on the properties that is useful for multi-objective molecular design. Here are some papers that is mentioned in this talk. Just as a last slide, I want to mention that our recent paper, that we are really trying hard to make these machine learning models really used for drug discovery. So here we just had a new paper accepted in Cell called Deep Learning Approach to Antibiotic Discovery. It is a Cell article paper, a Cell cover article. We basically used property prediction techniques, and we tested molecule, and we found that using property prediction, we can find novel antibiotics that is experimentally validated to be to have a strong killing efficacy against a wide range of different bacteria species. So in the future, we also plan to apply all the generative models that I developed for real-world discovery tasks, ideally to validate them for antibiotic discovery. So with that, I want to conclude my talk, and I want to thank you all for listening to my talk, and I want to thank you for AISC, all the members, for helping me facilitating this talk. So now I'm open for more questions. Thank you.
Thank you, Wengong. We're open for questions. So if you guys have questions online, you can ask them through YouTube or other platforms, and we will transfer that to Wengong. One question online, they're asking, they're wondering how your motif extraction might work for peptide natural products that may contain multiple peptide bonds but few rings. I think we discussed this prior to the presentation. Yeah, go ahead.
Right. So how can I say, the motif extraction method, it utilizes some part of prior knowledge. So for these peptide compounds, what we can do is, for example, break some of the bounds and then see what are the substructure, what are motifs that occur frequently in the data set. I don't have a super concrete ideas for now, but basically I can, for example, as a brute force approach, I can enumerate, I can try to enumerate by these motifs by breaking some of the bounds in the peptide compound, and then see what are the motifs that are that occurs frequently. We also can use these property guided methods like Monte Carlo tree search to extract motifs that are correlated with some of the properties that you care about. I think this is a more principled approach that can that requires less prior knowledge that can be widely applied to different domains.
Yeah, so just to add a bit of the information with peptides and amino acids, you can based on this model, the model has the flexibility of being able to break the bonds in the places that you want. So with peptides, you can break peptide bond, which is the amide bond, and get the amino acids. You don't necessarily have to break the cyclic structures if I understood correctly. Yeah, Wengong?
Mm-hmm.
Okay. So
yes, definitely true.
It's actually, if you think about it, it's a simpler. It turns into a simpler problem than dealing with other structures. So let's see if we have more questions. Okay. So it seems like we don't have any other questions online. I'm going to ask you a couple of questions myself. If you go to slide number 29.
It's probably around here. Sorry, I don't have the slide numbers. Yeah, that's my. I think it's here.
It's a hierarchical graph encoder, bottom up structure.
Yeah.
Yes,
Yeah.
Exactly. So if you could please, for better understanding of the audience, explain to us how the nodes work in this process. Nodes and edges, because you're using the like transferring the information, and the structure is the graph structure. Could you explain what you're showing here?
Oh yeah, sure. So just to recall, this is the motif layer. Each node is a motif. This is attachment layer, and this is the atom layer. So there are different types of edges. Let me see. So here, for example, these dashed arrows represents how the messages from the atom layer will be propagated to the attachment layer. For example, after you run this message passing network on the atom layer, you will have these vectors associated with each atoms, right? So I will propagate these vectors to each of the nodes here, which is each motif. When you are running attachment layer NPN, each node will receive message from the node from its neighboring nodes here, and here, as well as these atoms belongs to these motifs. So it will receive messages from the atoms in all of these gray boxes. And you can do the same thing from attachment layer to motif layer.
Okay, so your nodes would be the motifs that you selected from the vocabulary. Okay, is that correct?
Yeah, that is correct.
Another question I had at the atom layer, is that basically the same or similar process as the junction tree work that you did previously?
Mm-hmm.
Okay, and then that the information is transferred to the other layers, which are basically message passing networks too.
So back then in the junction tree VAE, the junction tree layer and the atom layer is actually separated. So there's no hierarchical message passing from the atom layer to the junction tree. So
the junction.
Yes.
Yeah, the junction tree. What I mentioned was the junction tree seems to be just or has similarity to the atom layer only. Is that correct?
Yeah.
Okay. Let me see other questions. Okay. Slide number 36, experiment 2, de novo molecule optimization. If you can please go to that slide.
Yes.
I think it's the molecule optimization experiment 2. Okay, so here you're suggesting that you're going to use properties to optimize the structure. How are you introducing this information into your generation model?
I see. So this information is actually inherently provided in the training data. So here, if you consider this training set of source target molecules, you will have a starting molecule which has a poor, let's say, drug likeness, and the target molecule that looks similar to the input will have a high drug likeness, and that's how the properties is introduced into the model, and the model does not have direct access to the property predictor. It only knows what kind of graph transformations that will lead to that will typically improve the property. So in that sense, the model does not directly interact with the property evaluator, but rather observe them through the data that that model is being trained on.
Okay, so if I understand correctly, you're generating the models, feeding them to another model which predicts their properties, and then that will be a way of evaluating the how model generates those models. Is that correct?
That's correct.
Okay, that's great. Let me just double check if we have other questions. People are saying thank you for your presentation. One question, they say, you probably mentioned it, but could you elaborate again how you ensure that generated molecules are valid?
Right. So yes. So this is something pretty similar to junction tree VAE. First of all, all the motifs are chemically valid. So that means that each step, when we expand the molecule with one motif, we will always, stay within this zone that the output is always valid. When we attach this motif to the current graph, we will also make sure that it will not violate any of the chemical constraints. And to be more specific, when you attach this motif to the current graph, I will just use RDKit to check whether this attachment is valid according to the RDKit chemistry rules.
Okay. So another question that I had, if we're already labeling the atoms and we kind of know that the attachments, what's the likelihood of the model generating a structure that is not valid?
It's hard. It's very rare, but it's really hard for me to quantify. Because it's a very simple validity check.
Yeah.
Yeah.
Yeah. What I was thinking is that the likelihood would be very low. Let me just check what other questions are available. Let me see. Select the motif in G if its occurrence is the training. Okay. They're asking if you just filter out the ones that are invalid.
You mean filtering out during which stage?
This
is it like when you? Yes.
I think they're asking basically how you check the validity, because you're not filtering out any structures. It's just doing the validation test is later on, and that's done using RDKit, which will check if the structure makes sense based on chemical rules. So number of bonds, how they're connected, so on and so forth. So I don't think there's any filtering involved. Is there any filtering involved?
So maybe I didn't describe this very carefully. By checking the validity, I will check validity in each generation step. So to make sure that all the partial molecules that's been generated is valid. So if you have a motif and if the model happened to make a bad prediction, like attaching this into somewhere that is totally not right, I will just reject this prediction and we'll move on to the second best prediction. I do a greedy decoding when conditioned on this latent code Z. And so you can move on to the second best attachment prediction. Or if you're doing random sampling, I can just sample once again by rejection sampling.
Okay. Okay, they're saying that makes sense. Thank you so much. If the audience doesn't have any other questions, seems like we don't have any more questions online. I'm going to thank Wengong, and with that, we're going to end this session. Thank you, everyone, for joining us today.
Yeah, thank you so much.